For introduction, in plant biotechnology, the use of certain DNA sequence to detect genetic variation and support plant breeding and trait selection is known as DNA marker technology. A PCR-based method called RAPD, randomly amplify molymorphic DNA, amplify random DNA section using short randomized primer exposing polymorphs based on different in primer binding site. Rapid genetic analysis and comparison between individual or species are made possible by it. Next, RFLD is a method of DNA analysis that compares fragments generated by restriction enzyme dissection in order to identify genetic variation. Third is single nucleotide polymorphy, MS or SNP are individual different in a single of DNA receive a genetic marker for diversification and may be researched. SSR simple sequence repeat is a brief repeating sequence of 1 to 6 DNA base pair. Next is principle of marker technology in plant biotechnology. First is extracting DNA. Isolating high-quality genomic DNA from plant tissue, including leaf or seed, is the initial step. The template for additional molecular investigation in this DNA. Poor DNA is obtained using a variety of procedures such as commercial kits and ctDNA. Next is quantification of DNA and quality verification. To make sure the extract DNA is appropriate for amplification or ligation, its integrity is examined using agarose gel electrophoresis, and its quantity is measured using spectrophotometry such as nanodrop. Third is amplification. For PCR-based marker, for instance DNA, segment containing target areas are amplified using primers specific to RAPD, SSR, and SNP. DNA is sliced at precise recognition location by restriction enzyme for ligation-based marker, such as RFLP. For the fourth is gel electrophoresis. Agarose or polyacrylamide gel electrophoresis is used to separate the amplified or digested DNA fragments according to size. It is possible to employ capillary electrophoresis technique for high resolution separation, particularly for SSR or SNP. 5 is detection. RFLP via hybridization with type probe, either radioactive or non-radioactive, and certain blotting SNP using sequencing technology or detection technique based on fluorescence. Lastly, data analysis. Variation in nucleotide fragment, lack or the presence or absence of certain genes are plotted and examined to find attributes, assess genetic diversity, or create linkage maps. Polymorphic patterns are employed.
Application of the technology in plant. First, marker assistance selection must. Using molecular marker, this approach selects plant with genomic error implicate in the expression of desired feather. For instance, new rice variety will become more resilient to abiotic stress. Second, mapping gene tagging. Locating crucial gene on the plant genome is aided by DNA marker. For example, all of the major mapping family resources that have been generated since then have been based on traits and molecular variation among interfetal indica, japonica, and javanica subspecies. Third, identifying plant varieties. Plant species or variation can be distinguished from one another using by DNA marker. By identifying the smallest genetic variation unique among individuals in a plant species such as B. allelic variation among individuals that takes place in gene, promoter, exon, or intron. Conclusion. In summary, the future of modern agriculture depends on the rapid development of superior plant varieties made possible by DNA markers. This will help provide food security and sustainable agriculture in the face of climate change.